We're going to take a look at hyaline cartilage, and we're using the example of the trachea, and I can also take a look at the epiphysis of a developing long bone. So as always, let's kind of click back over to our slide box, Chapter 5, Cartilage and Bone. I'm scrolling down here and I chose trachea, and then we have our articular cartilage, and we can see pretty much right away that this is developing long bone, and we can go up to the epiphysis area and take a look at the remaining cartilage that is still there. So here we are with our tracheal area. This is a little bit of a different section. Yours is going to be more like a cross-sectional cut of the trachea, as if somebody went in and kind of just took like a tray like out of your trachea. And so you get this kind of like C-shape cartilaginous ring. If you were to kind of cut more of a longitudinal section, you can actually feel the little rings of cartilage that continue down your throat area. The video of me is gonna get eliminated because I need more of the screen at this point. So let's go ahead and take a look at what is more typical to what you guys are seeing in the lab section, which is this bottom view here. Your slide actually is the tracheoesophageal slide, and so you actually have two different rings of tissue on the slide. And so I usually say to students, put the slide on a piece of white paper and go ahead just with, the naked eye, look at what you're seeing. You should see these two rings, and one is going to have almost like a much more of a circular nature to it. They both are circles because one's at the trachea and one is the esophagus, but you got to identify which one is which. And then you have like connective tissue that's going to join the two of them together. So look for this kind of solid material, which might not be this dark on your slide really, but you're going to have this solid material that's forming some sort of a C shape on the slide. And so try to go ahead and focus in on that one so that we can look more at this cartilage area here. Now, in the future, we're going to go into the respiratory system. We're going to go into the gastrointestinal system, and we're going to look individually at the esophagus and the trachea and all of the fun tissues and structures and layers that are involved with those two tissues. But really for the purpose of, this video and what you guys are looking at in the lab, we're really just trying to get in there and look at the cartilage, right? And so as we're zooming in, try to keep your bearing, right? What is the outer peripheral surface of the trachea and what is the tracheal lining? And this is the area where air would be essentially kind of flowing through. You can also start to see color differences, and you can see the lining of the trachea. So this darker purple line here is going to be that pseudostratified ciliated columnar lining that you guys looked at before. You also have a little bit of connective tissue that's in here. You might run into some glandular areas. There's going to be possible muscle, fat, and this is another great area to find blood vessels. So you can also find different sizes of our blood vessels from arteries and arterioles all the way to veins and venules as well, and obviously capillaries. But again, let's try to focus on our cartilage. And this is kind of the difficult part and the fun part of these tissue slides, where it's like you have you have your trachea, you have esophagus, because there's so many other things that we can see on that slide. It's not just like skeletal muscle. So we start to see this kind of thick pattern here, right? And we know with cartilage, it takes on this kind of watercolor wash background for the matrix, right? There is still protein fiber in this matrix, but we don't necessarily always stain to see that. So as we're getting closer and closer, again, that watercolor background is really kind of apparent, right? And on your slide, this might be more of a lighter purple, not maybe so intense as we see here. You have these very large white dots everywhere, which are really kind of telling us that these are where the cells would be located that actually made all of this lovely cartilage tissue. And those white spaces are going to be the lacunae. Now, we don't see every single cell with every lacunae because, again, this is a flat section, right? We don't get to look at this in kind of a 3D format of actually going through the entire ring of cartilage. So you're going to get little snippets of maybe the surface of the chondrocytes or the surface of, let's say, the lacunae in some areas. So these kind of darker areas, that's exactly what that is. You're at the kind of the surface of before you would actually jump into that lacunae with the actual cell itself. As you get to the periphery, a lot of students will say, well, is this where I would find those chondroblasts, right? And chondro is cartilage, blast is a cell that is actively making the product, right? And actively making cartilage in this case. And the site, the chondrocyte, again, a cartilage cell, but it's more in an arrested, mature phase. And that's when we would see that the cells have a lacunae around them. So would the blast be out here to the periphery? Well, if this was a developing section, then yeah, that's where you would find the chondroblast adding cartilage to the periphery and to the outer surface. But because this is an adult tissue, and you can see that the lacunae go right to the surface, right? And both sides, that we do not have chondroblasts at this time. Not to say that there's not a few in there, but primarily this is all going to be adult tissue. So even if we zoomed in a little bit farther here, we can see, again, this nice background of our cartilage matrix, nice and smooth. The open white chamber is going to be the lacunae, and every so often, as the tissue is cut, we can find the actual chondrocytes inside, right? They're living inside of that tissue. As we go out to the periphery, if we go either that direction or this direction, you'll see that there is this surface area of a color change. You start to see it changing back into kind of this pink color, which tells us that this is going to be more of just the protein fiber. Now, protein fiber is in here too, right? We know that there's collagen in here, but we're not typically staining to see the fiber. It's in there. But out here we can. So there is that difference of cartilaginous matrix versus something that is more like a dense irregular connective tissue out here. So this is where we start to run into our perichondrium. And what is nice on this slide also is that we have those listed here. So we have what's referred to as the outer fibrous and the interchondrogenic layer. These are the two layers that make up our perichondrium. Now, we have to remember that not all cartilage and bone surface has kind of membranous covering. So where there's going to be any sort of wear and tear or two surfaces kind of rubbing up next to each other, or really a mobile surface, we're not going to have, like, let's say a perichondrium or an outer bony layer, right? That will be there, like a periosteum, right? So this is not that case. This is the surface, right, of the cartilage on both areas. This isn't a joint area. So we would find a perichondrium on both areas. So if we even click on this, let's see where it takes us. All right to that connective tissue surface. And again, look at all of these lovely chondrocytes, right? We also get into the outer fibrous, which is going to be where we find lots of fibroblasts. So all these little dark purple staining nuclei are telling us that these are fibroblasts that make a lot of the dense irregular connective tissue that surrounds the actual cartilage that is here. And so even if we click on that, it just zooms in a little bit more to basically say this is where you are. You might also have little blood vessels and things out here. And then the interchondrogenic layer is really this particular layer here, right? It's has fibroblast-like cells, and they can essentially make both collagen fiber, which would be collagen type 2, or they can actually differentiate more into a chondroblast, which is really interesting. So you have these borderline cells that kind of act like adult stem cells, don't they? If they can, but this is, again, still all connective tissue. So whether we're talking about dense and regular connective tissue up here, or let's say some cartilaginous material, we have collagen in both. And then we also have the ability to lay down a little bit more of that cartilaginous matrix at the surface. So if there is, let's say, any sort of damage or trauma that occurs, these cells can basically be activated to hopefully repair some of that. We know that cartilage isn't well skilled in repair because, again, we don't have blood vessels traveling through this tissue. So if I zoomed out, you can see, ooh, that's too far. You can see the absence of blood vessels, right? There's no blood vessels in here, none, right? And out here, we have blood vessels out here in the fibrous layer, and also out here. So we can really start to repair some of the collagenous fiber. We can repair fatty tissue. And we also have, if we go towards the lining, we'll see again that cellular layer, and then we have the chondrogenic layer, and then that runs right into maybe some glands we'll look at later in the term. And we also have some more connective tissue, and then that's when we run into our obviously lovely lining, our pseudostratified lining that is here. And so you can identify, again, the perichondrium on either side of the cartilaginous section of the trachea. And the same thing would happen with, let's say, like the ear cartilage for the elastic. And again, we have that inner chondrogenic and then the outer fibrous layer that is here. Hyaline cartilage is pretty typical in appearance of what we're seeing here. It really does look like this, even when we head over to, let's say, the epiphysis, right, of a long bone, and you have that articular cartilage that is there. It's very similar in appearance to what we see here. All right, so nice, lovely, kind of uniform chondrocytes, so they're really nicely evenly distributed. When we look at elastic cartilage, they're really, really tightly clustered together. And there's like not a lot of, there's matrix, but there's not as much matrix as we see here. So this is kind of the even distribution of cells. We have a kind of a medium amount of cartilage matrix. And then when we go to, let's say, fibrocartilage, lots of matrix, really dense matrix. And the cartilage cells, the chondrocytes, kind of are these what we call isogenic or isogenous clusters of cells. They kind of like island together, and so they're not as spread out as we see here. So one thing that we can do, let me give you this a pause. I'm going to head back over, well, maybe I don't have to pause. Let's see how fast we go. So if we go over to articular cartilage, and I have also a video for bone development as well, we can go to that articular cartilage here. And so you have a slide box, and your slide box, your slide says the fetal pig joint area. That whole epiphysis is still hyaline cartilage, so that would be another area you could go to. You could go to your epiphyseal growth plate slide, and it would have both primary and secondary ossification. So this bone is basically kind of like in development. But again, this whole area here is cartilage in kind of that growth and development phase. So you could literally go to the side area here. We wrap up all the way around and all the way back down. So everything that is purple here is hyaline cartilage. This is in development, and we know that because this is still cartilage. So once this is a mature bone, this will all solidify into bone material, and the growth plate, basically, the active growth plate stops. So let's kind of ignore maybe this area. I can even maybe go on to the side and show you that transition. So you get to see all of the growth zones here for our endochondral bone that we would take a look at in another video. But again, you see kind of really active formation of hyaline cartilage here, and then we're getting more of a transition over here with growth and development in a bone. But as I kind of click in here, again, you can see that lovely even distribution of chondrocytes, lots of kind of uniform matrix. Lots of these have lacunae, which would be adult cartilage cells, right? So those are chondrocytes. And then if you were moved to the periphery, this is kind of fuzzy, but you can see that these would essentially be our chondroblasts because they are at the periphery, kind of continuing to make bony material, and they don't have any lacunae around them. So let's kind of zoom back out and let's go up here a little bit and let's see what we find. Let's pull this up a little bit. Let's zoom in. Okay, so you can again see there's a little bit of a matrix color change happening, and again that's because of growth and development. So at the periphery here is where we would find our continuing growing, let's say, cartilage in maintaining of cartilage. And then as we head towards the center of the epiphysis, we can see that there's an obvious change of cartilage basically making trabeculae of the spongy bone. And again, all of these cells with lacunae are going to be our chondrocytes. And I wish it wasn't so fuzzy. Let me see if I can travel around. Not so much. And I apologize for that. But out here would be essentially your chondroblasts. So if you're in the lab section, go ahead and take a look at the periphery and see if you can find chondroblasts that are there. And again, the absence of lacunae. So maybe if we travel around, let's see if there's any that kind of come into a focus. Again, those have lacunae, so I would not call those blasts. Let's travel all the way down the other side. Maybe up in here. So again, I apologize that I can't, unfortunately, zoom in and kind of find focus to our own vision. But that's essentially what we got. Over here, it's just lots of mesenchymal cells giving rise to all different types of connective tissue. Remember that mesenchymal cells are mesodermal in nature and can give rise to all of our different types of connective tissue, which is pretty great. Trying to think of any other area. Maybe I can jump down there. And even a lot of those look like chondrocytes too, right? And then we get more into kind of our fibroblasts over here on the side. So the growth plate area, which I kind of want to stay away from, you can possibly see some chondroblasts like up in this area. This is pretty further into development, but kind of the border between, let's say, the resting cartilage and then the bone developing at the epiphysis, you might be able to see some of those chondroblasts here. As you get more towards the center, they really don't exist. Even the resting cartilage almost completely dissipates. So we can talk about that with growth and development. So hopefully that's helpful. You got lots of slides to choose from in your slide box for hyaline cartilage, anything from the trachea to the fetal pig slide to this particular slide as well. And anything that's really kind of bone development, you will be able to see cartilage developing, specifically hyaline cartilage. All right. Thanks, guys.